Okay, so if you're playing along, I hope you are, following along in the slides. If you're following along in the slides, you see we are at a slide where we are going to be talking about the cell membrane. So when we talked about a cell is bounded on the outside by a cell membrane, and it has structures on the inside that we'll be talking about also. So first thing we've got to talk about is that outer limits to the cell, which is known as the cell membrane. Sometimes you'll hear it referred to as the plasma membrane. Now, when we were talking about biochemistry, I know you're like, oh my God, I thought we were through with that. No, we'll be touching base on that off and on for the next 9 months. But anyway, when we were talking about biochemistry, we had talked about this funny little molecule. We had talked about a triglyceride. That's not the funny one. We had talked about a triglyceride, and we had said that's a fat. And what does it consist of? It consisted of glycerol, 3 carbons, and to each of those carbons, there was a fatty acid chain. And we had said this thing was nonpolar. It hated water, hydrophobic. Now, when we talk about the cell membrane, you might remember we had said there is a modification to that triglyceride to produce a phospholipid. So maybe you remember. And what we said is one of the 3 fatty acid chains is, instead of being a fatty acid chain, it is replaced with a phosphate group. Now, phosphate groups are polar. And so we said that we got this polar thing that's attached to this non-polar molecule, the rest of it. And we said there's a specific name for a goofy molecule like that, and it was called amphipathic. So why is it goofy? At one end it's polar, at the other end it's non-polar. That's unusual. My email keeps going off. So we had this phospholipid, and you might remember we said cell membranes are made of them, and we had said when you put this molecule in a liquid environment, what happens is the phosphate portion, it likes water. It's polar, right? It's polar. Water is polar, so it likes the water. But those fatty acid chains, they don't like water. So what we said happens is you have one phospholipid up here and one phospholipid down here, and they organize themselves in sheets. Now, if you look at the picture on the PowerPoint that I've got pulled up over there, that I'm going to put this at the bottom of, you will see that we have a phospholipid bilayer, and it's drawn in a much better manner than I could draw it when I was putting it on my little whiteboard. And so you can see it's like a sheet, and all these phospholipids are organized with the phosphates up and down. And that's because there's water on the outside, water on the inside, water everywhere, because we're 70% water. So water is everywhere. So what happens is these things organize themselves so as to get the fatty acid chains as far as possible away from the water, either outside or inside. And it ends up, it's a pretty good barrier to everything, right? So if the molecule has polar portions on the outside and non-polar portions on the inside, anything trying to come through there is going to encounter something that it's not comfortable with. And so this phospholipid bilayer is a great barrier. Now, if you look at that picture over there, you say, well, I see that orangey phospholipid bilayer. There's a whole lot of other stuff in there. And if you look at them, those little sort of purple or bluish, blue and purple things over there, those are proteins that are embedded in that phospholipid bilayer. And it ends up Now, I want to talk about the phospholipid portion just one more minute. What is it like? What is, how, when you interact with it, what is its characteristic? It's like a soap bubble. So if you got the bubble stuff, right? You got the bubble stuff and you blow through the little loop and you make these little bubbles, and maybe you're really fascinated with those bubbles and you're watching two of them and they're floating around and they come together and they kind of stick and then boop, they become one. So that's what that phospholipid bilayer is like. It's very fluid, and it literally can combine with another phospholipid portion. So in addition to that fluidity quality, we have these proteins that are in there. Maybe you love art. Possibly you're an art lover. And if you are an art lover, you would know what a mosaic is. Like the Romans have these beautiful pictures where they take these little colored things, they stick them together, and you've got a Roman gladiator there. Well, scientists are nerds. I admit it. Scientists are nerds, and so they looked at this and they decided that those proteins that are in that liquidy phospholipid bilayer, why, it's like art. And so you can make a puppy. No, it doesn't make a puppy. But they made this analogy. So when they describe the cell membrane, they came up with a model for it. And the name for that model is the fluid mosaic model of the cell membrane. And it's making reference to those proteins that are sticking through there like they're art. They're not art. They're proteins. They have very specific functions we'll be talking about on the next slide. And so on here it says it's the outermost limit of the cell and it regulates the passage of materials into and out of the cell. We said not a whole lot can pass through a phospholipid bilayer. Water can actually squeeze through there on occasion, but it's not very efficient. So we have other mechanisms for getting materials into and out of the cell. And the way we do that is by those proteins. Okay, the one other component of the cell membrane that I haven't mentioned yet, but if you look carefully at that picture, you'll see it, is cholesterol. We always think of cholesterol as being bad, and it can be, but it's not bad. It actually gives rise to some hormones, and it also, in this case, it helps to make that fluidy phospholipid bilayer a little bit less fluid. It's still fluid, but it stiffens it up a little bit. And finally, before we go on to the next slide where we're going to talk about the function of all of those proteins in there, we are going to, I'm going to read a quote, and I'm going to talk about the carbohydrates that are sticking out there. So I have just a little bit more. The fluid mosaic model, a description of it is a mosaic of proteins floating like icebergs in a sea of lipids, a constantly changing mosaic of phospholipid, cholesterol, and proteins. Now, just to make reference to COVID-19 and the viruses, I said COVID-19 will bind to a receptor, and those proteins can be, some of them are functioning as receptors, and that was the final point I wanted to make. I better stop here. I've gone on too long.